repair is that here the portion that containing the damaged bases, that is totally cut off from the strand, and new strand containing the correct bases, that is attached with the or pairing with the another strand. Okay. So here the main important thing is that how this is happening. The repair system includes the breaking of the phosphodiester bond on either side of the lesion. Okay, the phosphodiester bond is cut off from the two part or two sides on the same strand, not the both strand, only the strand that containing the damaged bases. This is cut off from the both sides. Okay, and this excision leaves a gap which is filled by the two enzymes functions, that is the ligase and the DNA polymers. Okay, so here this is an example is that UVR system. This UVR system, this is a great example of the excision repair, and it is found in the E. coli. Okay, so here